Please let me know if you can see the slides.
Yes, we can, but they're not full screen yet.
Okay, yeah, let me just make them full screen and you
We
should
can
be able
make
to see
full
them now.
screen. Perfect.
All right, awesome. Well, thank you very much for the invitation. I'm really sorry I wasn't able to make it. I got COVID last week. Sucks. And doing much better, but have this lingering cough that's very annoying, so I apologize in advance for that. Also wanted to say congratulations to Martin. We're very happy for you. And the purpose of this talk is to go over a case study on the cases of male reproductive toxicants. Um, but also wanted to give some highlights of some of the work that's gonna be coming up next. Before I go ahead, this quick disclaimer: the views expressed here are those of the authors, myself and my colleagues, and they do not necessarily represent the views or policies of the US EPA. I wanted to do three things with this talk. Just give a little bit of background on the cases of male reproductive toxicants, talk about this case study using benzoylpyrene, and also give you a quick snapshot of some of the work that we're just getting started with and next steps for the cases as we're applying them at EPA. So I work, pardon me, in the Office of Research and Development within the EPA. And most of my work is focused on IRIS toxicological reviews. And as many of you know, IRIS tox reviews can cover a variety of types of toxicities and may include many dozens, if not hundreds, of epidemiological, toxicological, and mechanistic studies. This can become challenging to dive in and analyze a large number of studies and do so in a transparent way. In the case of toxicological studies, one of the challenges is the complexity of the database and diversity of experimental design models, types of outcomes, species, strains of animals used, etc. And one way by which we address this complexity is by developing what we call evidence inventories. And in an evidence inventory, we'll collect information um, from the studies identified in our systematic review, and things like citation information, but more importantly, experimental design features such as test compound, age and life stage of animals used, dose, route of exposure, animal species and strain, etc. And then of course, the types of outcomes reported in these studies. In the case of reproductive studies for male and female, we rely a lot on our own reproductive guidelines and its description of outcomes that are commonly evaluated in and reported on in reproductive toxicity studies. And the table that you have here is pretty much taken verbatim from the EPA reproductive guidelines. So that's great for doing an inventory that is focused on toxicological studies, but then to do so an inventory on mechanistic studies, that also can pose a challenge because we don't have this sort of categories, if you will, to organize the available evidence in the peer review literature. Early on, when we were developing these studies, these inventories, we were just tagging everything as mechanistic, and we quickly found out that that wasn't going to work. Around that time, I was working on an inventory for PCBs and focused on male reproductive toxicity and having this challenge. How do I organize all of this mechanistic evidence that I'm trying to capture in the inventory? And two of my colleagues, Catherine Gibbons and Vince Cogliano, mentioned the key characteristics of carcinogens. And honestly, after reading the paper, I felt like just using the 10 cases and applying them to this inventory or database that I was trying to generate. But we also realized that it was not specific to male repro, that there were some things that we were missing or not capturing quite well. So we started to draft our own set of what we were calling mechanistic categories. At the same time, we found out that Martin and others were organizing a workshop intended at extending the concept of the key characteristics to non-cancer outcomes. And for that workshop in 2018, we were focusing on endocrine disruptors as well as reproductive toxicants. Sorry. So we were able to get ourselves invited to participate in that workshop, and we really appreciate it. And the result of that workshop was the identification of the 8 key characteristics of male reproductive toxicants. And these are including germ cell alterations, somatic cell alterations, changes in hormone production or levels, changes in hormone receptor or changes in hormone receptor functions or levels in the cell, genotoxicity, epigenetics, oxidative stress, and inflammation. So this was all really great to have them. The next step we felt was to try them out and perform a case study, to conduct a literature search, do our screening, develop inventories, etc. And for that, we chose benzoate-pyrene. So we performed a literature search focused on BAP and male reproductive toxicity, looking at toxicological and mechanistic studies. And the goal was to apply the KCs to screen the studies that resulted from that literature search, develop evidence inventories, and do our evaluation of the mechanistic and toxicological evidence. Here's a quick breakdown of the literature search that was performed on BAP and male repro toxicity. So we identified over 2,000 citations from several different databases. Then we used systematic review software tools to perform our title and abstract and full text screening. We narrowed it down to 64 citations that we included in our inventory. The current number is actually a little bit larger than this. It's close to 80 studies, and that's because we've been doing some literature updates, and we recently did one and identified some new studies that we're adding to our inventory. Once the inventory is developed, the nice thing about it is that you can use it to navigate through the available evidence and do a deeper dive. And here I just break it up by according to the KCs and the number of studies that inform each of the KCs that are currently captured in the inventory. One aspect that I wanted to spend a little bit more time on is the KC for effects on somatic cells. And we identified 22 studies informing this KC. But there's also a lot of large diversity of endpoints captured within here. Things from like cytoskeleton changes in Sertoli cells, apoptosis in Leydig cells, biochemical changes, molecular changes, cell signaling aspects. And it was a little bit still kind of tedious to navigate through that. So we wanted to do something that it's a little bit more nuanced. So in the end, we borrowed from the KCs of female reprotoxicants, and I'll tell you more about that in a moment. Next, I want to show you a couple of examples of the information captured for each of the KCs in our inventory, and this is just a couple of them. So for oxidative stress, we have studies using both in vivo and cell culture models, the majority of them reporting an increase in ROS production, as well as markers of oxidative stress in different cell types and parts of the male reproductive system. Another aspect that we tried to capture in our inventory are intervention studies. And what these studies report is that intervention with antioxidants or aryl hydrocarbon receptor inhibitors, as expected, ameliorated some of these effects. Here's the information for the KC on hormonal effects. And again, the inventory captured studies both in vivo and in cell culture using different research models. And again, the majority of the studies report a disruption in steroidogenesis, specifically androgens, after exposure to benzoate-pyrene. And again, the intervention studies also report that intervention with antioxidants or AHR inhibitors ameliorated some of these effects. And here's the KC that we borrow from the list of female repro KCs for signal transduction. So we incorporated this to our inventory. And the way that it helped us is to capture some of those cell signaling cascades activated early on in the process after BAP exposure, specifically activation of AHR and the cell signaling that's downstream of AHR activation. Sorry. Another aspect that I wanted to mention about applying the KCs is that as we dive deeper into the inventory and organize it and according to the KCs, the type of studies, species, strain, etc., we start to identify different mechanisms or components or pathways that by which BAP causes toxicity. And as we do that, we start to identify the linkages between these different mechanisms and how they associate with each other. Also, it's worth mentioning that this is not that different, and in fact, is compatible to the analysis that was done previously in the IRIS toxicological review of benzoyl that also considered mechanistic evidence on BAP-induced male reproductive toxicity. So what I'm trying to say is that the KCs and other frameworks such as MOA can work with each other and be compatible with each other, as we're finding out here. So in conclusion, adding the KCs to this process definitely improves transparency, and it definitely helps with navigating all or most of the available evidence that we identified in our systematic review without me having to remember, oh, what was that paper published in 2004 that used Sprague-Dawley rats and reported on oxidative stress? So once it's in the inventory, it's a lot easier to navigate to that. And then lastly, I wanted to mention quickly some of the work that our two postdocs, Dr. Bevan Blake and Timothy Anderson, started working on and expanding on the KCs. So they're trying to build confidence in mechanistic evidence derived from high throughput screening assays that is used for hazard evaluation. More and more, we're considering NAM data or new approach methodologies in our hazard evaluations. However, integration of this type of evidence to frameworks such as MOA is not well characterized. And what they intend to do is to use the KCs as a basis to appraise the information that is currently available in ToxCast. Specifically, what they intend to do is to determine the extent to which in vitro bioactivity data, so from high throughput screening assays in ToxCast and Tox21, informs the KCs of male and female reprotoxicants. They will also leverage the BAP case study that I just described to further explore integration into of ToxCast and Tox21 into MOA analysis. And then finally, they would like to interrogate the extent to which the NAM data in ToxCast and Tox21 support weight of evidence evaluation for developmental neurotoxicity. That's my presentation. I want to say thank you to my colleagues at EPA who participated in this. Also, the KC's work group. I would really appreciate the opportunity to participate in this process, and also my colleagues in the EPA Tox Pathways work group. That's it. This is my contact information. If you have any questions, comments, I'm happy to continue this conversation.
Excellent. Really thorough and insightful talk. Thank you so much. And everyone